Okay. Thank you very much for this introduction. So thank you for inviting me. It's a great pleasure to be in Vienna, and I got to talk to many of you today. So that was very nice, very interesting. So yes, so I am working in a lab that is, sorry, I cannot see my, oh yeah, sorry, it's here. So working in a lab that is quantitative genetics and evolution in Le Moulin, which is a lab located in the Université Paris-Saclay, which is south of Paris. And I'm going to talk to you today about, so I changed a bit my title, but I'm going to talk to today about the wild domestic tango. I thought it was kind of cool to illustrate that I'm going to talk about phenotypic evolution, but also two-way gene flow between wild and domestic forms. And so I first want to start by thanking the funding, so the ANR, INRAE and Région Bretagne, but most importantly, to thank two PhD students that I'm going to present their work today. So the first one is Ewen Burban, who defended some time ago and developed a tool that is called REACH that I will present. And Arthur Wojcik, who is still doing his PhD, he's going to defend next March. He will be looking for a postdoc. And so all the work I'm going to present today is basically the work of these two PhD students. And I took a picture here in Mexico some years ago. And this picture illustrates an ear that has a phenotype that is a bit intermediate between a maize and a teosinte, so the direct ancestor of maize. And so just to illustrate that there is some gene flow between maize and teosinte. So first of all, what is domestication? So domestication can be defined as an evolutionary process whereby a population adapts through selection to new environments created by human cultivation. So everything basically starts with wild population evolving in their natural habitats under natural selection. And sometimes in the past, those wild populations were put into cultivation. They were domesticated. So that occurred around 9,000 years ago in Mexico. And they acquired a number of traits to produce these traditional land races that you can see in the middle picture. So that's on the left, you have a wild teosinte population, and on the middle, a traditional land race. And after this domestication, the crop is bred and dispersed out of the center of origin, producing a broad range of traditional land races, where you can have an illustration of some of them here. And basically, those three last steps, cultivation, domestication, breeding, and dispersal, occurred under both natural and human-driven selection. So I told you that, okay, during domestication, plants acquired a number of traits that are profitable for humans, for human needs. And these traits are collectively referred to as the domestication syndrome. So this syndrome includes more compact architecture. So this is illustrated here in sunflower, where you have the domestic form that has only a single stem here, while you have multiple stems here in the wild sunflowers. And here in foxtail millet is the same story, right? The domestic form on the right compared to the wild form on the left. You also have a lot of means of dispersal. So this is illustrated here in rice. So you see that in the wild form, the seeds, they shatter at maturity, while in the domestic forms, they remain attached to the ear. And you see that the wild forms also have quite a bit of ornamentations that facilitate their dispersal by the wind or in the fur of animals, for example, in the wild environments. And in the domestic form, you have a drastic reduction of these ornamentations. You also have an increase in the size of the organs that are consumed. So that's illustrated here in tomato or in the root of the sugar beet here. And also a loss of defense mechanisms. And so this is illustrated in eggplants, where in the wild forms, you have some spikes here that have completely disappeared in the domestic forms. And this also applies to chemical defense. In addition, an important trait is the reduction of seed dormancy. So typically in the wild, the seeds are dormant and they don't grow during the bad season, right? But the domestic forms have lost or partially lost this seed dormancy. So of course, domestication has been a major, if not the major, transition in recent human history. It has accompanied the sedentarization of human population and their expansion. So this is a graph of the density of the human population in South America. So you see that, and here you have the years on the x-axis. So you see that it's increased, it's reaching a plateau. And during the Neolithic transition that included this start of agriculture, you have a drastic shift in the slope here. And that corresponds to this human expansion that led to the absorption and or the replacement of hunter-gatherers. And the domestication was also accompanied by greater technological developments, but also a greater labor division and higher social stratification. And more importantly, perhaps the most important for us right now, is that domestication has drastic consequences on our landscapes, on our environments, and also on the emergence of infectious diseases that is often linked to our close proximity to farm animals. So importantly, domestication has also established a mutual dependency between humans and plants. So domesticated plants are maladapted to reproduction and survival in natural habitats. So I just illustrated, for example, domestic plants that don't lose their seed anymore at maturity, which makes them really maladapted to reproduction and survival in natural habitats. But us as humans, our reproduction and survival also depend on a few domesticated species. So for example, this is a recent estimate from the FAO that shows that 9 plant species provide 75% of the human food worldwide. So when did domestication happen? So for plants, the oldest domestication that dates back 12,000 years ago, more or less, and it covers gradients of dates. And you see that the domestication of animals occurred more or less simultaneously, except for the dogs, for which it seems that the domestication occurred much earlier than for the other animals. So now, why is domestication a model for evolutionary biology? So domestication has been proposed as a model by Charles Darwin. So he actually wrote a whole book about it called The Variation of Animals and Plants Under Domestication. And why is it a good model? Because in a way, you have a proxy for the ancestors, right? So the wild plants that have, for most crops, still persisted in their natural habitat. So you can still find them today. And you can compare them to the actual organisms, the crops that are derived from them, that have evolved from them under the combined human and natural selection. And so this process, so here you have, the wild population. So they evolve under natural selection in their natural habitat, sometimes in the past. Some of these wild individuals were taken to be domesticated. So usually domestication is associated with a shrink in population size, okay? And then you have now a population evolving under human-made habitat, your crops, right? And the interesting things is that crop and their wild relatives, for most of them, or for a lot of them, they continue to exchange gene flow. So it's a process of divergence with gene flow. And again, you have in your hands thousands of generations of evolution, and you can compare the proxy for the ancestors and the derived plants and ask a number of questions, right? So these questions can be related, for instance, to history and anthropology, but you can also ask questions about the dynamics of adaptation, about phenotypic evolution. I showed you there are many traits that have evolved, about the genetic determinants of these phenotypic changes, about the molecular syndrome, for example, the syndrome that is associated to this shrink in population size that I described earlier. You can also ask questions about the crosstalks between these forms that still exchange gene flow. And something that I had a growing interest for is the impact of domestication on the interactions. between plants and their abiotic environments, but also plants and their biotic environments. And in particular, I have some research work going on the impact of domestication on plant-plant interactions, even from different species. But today, I'm going to focus on two questions: phenotypic evolution and crosstalks between forms. So first, phenotypic evolution under domestication. So to address questions about this, we designed a sample encompassing 13 diploid species that are shown here in pictures, and that are covering a gradient of domestication timing. So you see the most recent domestication, so like the one of apple, and more ancient one, like the einkorn wheat here. And these species also cover a diversity of life cycles from annual, biennial, and we also have perennial species, the grapevine and the apple. We also have a variety of baiting system, outcrossers in pink and selfers in green. So for each of these species, we sampled 20 wild individuals from 20 wild population and 20 domestic individuals from 20 traditional land races for each species. And we sequenced those 40 genomes per species. And so we, first of all, determined the genetic diversity in the wild and domestic gene pools. And what this x-axis is showing you is the percentage of wild diversity that has been retained in the crop. So you can see that for most crops, it's below 100%, meaning that most crops have lost diversity through domestication. This loss can be pretty severe, as in the case in foxtail millet. But you see that there are two exceptions, and those exceptions are apple and grapevine, our two perennial species. So then what we did is that we phenotyped a number of traits from 11 to 57 traits in each of these species, often in 3 replicates to control for environmental effects. And in this diagram, you can see the number of traits that were measured for each species that is proportional to the size of the circle. And you have the traits connecting the species that where the width is proportional to the number of shared traits that were measured between 2 species. So after we did this, we performed a detection of domestication-related traits in each system. So let's take, for example, foxtail millet here. We measured all these traits. For all these traits, we determined the mean and the variance for the domestic form in blue and the wild form in green. And then we run some statistical tests to see whether for each of these traits, there was a significant difference between the domestic and the wild forms. And when there is a little star here, it means that this trait was indeed significantly different between the wild and the domestic forms. So we did the same for einkorn, for all our species. And then here I plotted the exact same diagram as before. But here the size of the circles are proportional now to the number of domestication associated traits, and the number of domestication traits that are shared between species is represented by the width of these lines here. So we tested for convergence by resampling and found that domestication traits are shared by 3 or more species are more common than expected. So in other words, we found statistical evidence for convergence of traits, meaning that there's a tendency of the same traits, of same set of traits to have been selected over and over in different species, even very distantly related species. Then we wanted to determine the phenotypic evolution in a multivariate space, okay, accounting for all these traits. But we bumped into a first problem, which is basically we were trying to literally compare apples and oranges, right? So we measured a lot of different traits in species that are extremely different. And we bumped into these challenges of cross-species comparison because traits differ in their measures, in their scales. So to deal with this, we performed a multivariate analysis, factorial analysis of mixed data that allowed us also to mix quantitative and qualitative traits, because we also measured traits related to color of the flowers, and so really qualitative traits. So you see that when we perform such an analysis, for example, in maize, you're able to distinguish pretty well on the first axis the domestic form in blue from the wild form in green. And similarly, in common bean, it's even more clearer here in common bean, the distinction between the wild and the domestic forms. Now we run into a second problem, is that the samples, they really differ from one species to another. So for example, in one species, we may have samples, land races that are further away from the domestication center, so maybe more drift between the wild and the domestic compartments, or maybe the traditional land races have went through a greater level of improvement. So we really needed a control trait to make cross-species comparison to make sure that we could control for our sampling design in the different species. And this control trait needed the following features. It had to be easy to measure in all systems and reflecting evolution unrelated with domestication. So basically reflecting drift or even local adaptation or improvement, but something that was not related with domestication. So we thought about using the near-infrared spectra, right? So basically what near-infrared spectra does is that you basically illuminate an organ of the plant with infrared, and you get back the absorbance at different wavelengths, right? That is written in the form of a spectra containing over 1,000 wavelengths, okay? So if we do that, if we use this technology on seeds, which is an organ that is often targeted by domestication, and then you use the spectra to perform multivariate analysis, you see that you can distinguish, for example, here in foxtail millet, very well the domestic from the wild form. So this is because, you know, seeds, as I just said, is often targeted by domestication. So this is clearly not a good control trait because it's directly related to domestication. However, if you measure nears on leaves, which is not an organ, usually not really, we don't think about it as being directly targeted by domestication, you see that the difference completely disappears. So you can no longer tell the wild from the domestic compartments. So we thought this nears measured on leaves could be a really good control trait. So we verified that it had a biological signal. So for instance, we performed many technical replicates and verified that the technical replicates clustered together. And then what we did is we related the log ratio of nears measures on leaves in the domestic form over the wild form to our estimate of the genomic diversity in the domestic over the wild forms. And you see that there is here a correlation, meaning that this tells you something about drift. Okay, so that's exactly what we wanted. Okay, so we use this as a control trait. And how we did that is we computed what is called the Pillay trace for this control traits. So the Pillay trace is based on an AMOVA, right, run on a multivariate analysis, and it basically tells you something about the divergence, the phenotypic divergence. Okay, so we did this for the control trait. We call it Pillay control, and we also computed the Pillay trace on all other traits. And we developed this index that we call the multivariate phenotypic divergence index, which is simply the Pillay trace computed on all traits minus the Pillay trace computed just on this control trait. And then after computing this index, we ranked all our species, right, based on this index. So the first thing that we found is that all the species had a very high index, over 65, meaning that the divergence, the phenotypic divergence between the wild and the domestic forms is really high in all species. So the domestic space is not contained within the wild space. It's really well separated. And something else that we found is that this MPDI does not correlate with the domestication timing. So it's not because you have been domesticated a longer time ago that you have a bigger phenotypic divergence between wild and domestic forms. Then we wanted not only to to rank the species by their level of phenotypic divergence between wild and domestic forms, we also wanted to have an estimate of the phenotypic space of the wild and the domestic form. Okay, so in order to estimate this, we computed the determinants of the variance-covariance matrix computed on all traits, and we computed the ratio of these determinants. So if this ratio was below 0, it meant that the wild space, the wild multivariate phenotypic space, was greater than the domestic space. And if the ratio was above 0, it was the opposite. The domestic space was greater than the wild space. So you see that for most species, this ratio is below 1, meaning that the phenotypic space shrink during domestication in most species. And then when we relate this ratio to the domestication timing, again, we didn't find any correlation. And when we relate it to the loss of genetic diversity at the genome-wide level, we also didn't find any correlation with this ratio. So the conclusion of this first part is that, about the phenotypic evolution under domestication, is that basically it looks a little bit that this scheme that was proposed by Glemin and Bataillon in 2009. So here you have the ancestral adaptive landscape of the wild form. So this is a fitness landscape with two traits, dispersal and number of grains. So you have a peak here, the wild peak basically. And what happened during domestication is you have the emergence of a second peak, right, where you have less dispersal but also more grains. But what this figure doesn't show is that basically this peak, the space has shrinked, right, during domestication. This shrink is independent from the domestication timing and the loss of genetic diversity. And one possible explanation is that domestication traits are encoded by only a small portion of the genome, so selection on them does not correlate with the overall loss of genomic diversity. So now I'm going to my second part, which is about the crosstalks between wild and domesticated forms. And I will first I will first talk about it from an adaptive introgression angle. So maize was domesticated from one Teosante subspecies in Mexico 9,000 years ago, and this Teosante subspecies is Parviglumis. It occupies the green niche here that you can see on this map, and it was domesticated in the lowland of Mexico. But there is a second annual Teosante subspecies that is called Mexicana, that is shown here in red. This Mexicana is adapted to the highlands, to the highland Mexican plateau. Okay, and so here you have a picture of Mexicana, which, by the way, is known to be a weed infesting maize fields. So what the work of the team of Jeff Ross-Ibarra at UC Davis has shown is that if you sequence base genomes all over the Americas here, okay, you will find that 15 to 25% of the maize genome is actually of Mexicana ancestry. Okay, so meaning maize was domesticated from Parviglumis, but still 15 to 25% of its genome is occupied by Mexicana. And what is really interesting is that the Mexican ancestry proportion correlates with elevation. In other words, this is a good evidence that basically Mexicana, who was adapted to the highlands, right, contributed to the adaptation of the domesticated maize to higher elevation. So that's introgression from Teosante to maize, okay? What we also found is that the reverse is true. You can also have adaptive introgression in the other direction from maize to Teosantes. So this work has been conducted by the team of Valérie Lecor in agroécologie Dijon with Mathieu Siol and Christophe Dely, and I was just a collaborator in this project. So this project is about the French Teosantes that were first observed in the early 1990s. So Teosantes have always been known as endemic to Mexico. They were only found in Mexico. But it turns out in 1990s, people started to observe teosanthes in France, and they were later observed in Spain, in the provinces of Aragon and Catalonia, where they cause important yield loss. And what we first did is we genotyped those Spanish and French teosanthes along with some Mexican teosanthes. And the first thing that we found is that those French teosanthes are of Mexicana ancestry. So they come from Mexicana, not from Parviglumis. Then if you plant these teosanthes in Dijon, okay, you see that those French teosanthes, they flower at the same time as maize. But if you plant Mexican teosanthes in Dijon, they are not happy, right? And they flower very late in the season. They actually flower in the fall, where we harvest maize, so they are not able to produce seeds. So there is clearly here an adaptation of this French teosanthes to the European climate, right? Now they are able to flower under long days because our summer are very long days as compared to the Mexican summers. So how did they do that, right? How did they adapt it? So first, what we performed is the genome scans, and we looked at all the regions along the genome that differentiate the Mexican teosanthes, so the Mexicana, from the French teosanthes. And so the little red stars here that you can see along this chromosome 8 here are the potential targets of selection, okay? Then we use a software called ELAY to infer the ancestral allele dosage from maize to this French teosanthes. So you have here the French teosanthes in blue, and that tells you how much they are maize. And you can see that here there is a cluster of candidate loci that have been under selection. This cluster encompasses a gene that is called ZCN8, which conferred the early flowering under long days. And on top of this region corresponds to 2 allele dosage from maize. So the French teosanthes, they are fully maize in that region, right? Meaning that basically the gene flow from maize to these teosanthes have facilitated their adaptation and their capacity of flowering under long days. Another example here is in a gene called ACC1. So a herbicide tolerance maize variety is used in France since 2001, and it has a herbicide tolerance caused by a mutation in this gene ACC1. What the team of Valérie Lecor found is that 43% of French teosanthes have now become resistant to this herbicide. They are the RR teosanthes, and all the RR teosanthes, they are basically maize at this gene ACC1. Okay, and they sequence the whole gene in the French teosanthes and the maize, and there's not a single mutation difference. So clearly, there's a whole haplotype that was transferred from maize to teosanthes, so now they became resistant. So they have been following this teosanthe population for several years, and I was lucky enough to accompany them to the field last September. And so you can see here the teosanthes growing in the maize field. So here is Valérie and Christophe. And the teosanthes, you also find them in sunflower, in asparagus fields, in just natural or maybe disturbed habitats, but not particular fields, okay? And what we also found is hybrids. Sorry, it's difficult, yeah. So this is a teosanthe ear, classic teosanthe ear, and you see that from time to time we find these ear that are hybrids between maize and teosanthes, and this is where they are on the plant. So clearly there is some gene flow here. Okay, so now I'm gonna attack this from another angle, is the reproductive isolation angle. So there is clearly some gene flow between crop and their wild relatives, but there is also evidence for reproductive isolation. And this evidence come from, or maybe the best evidence, come from a meta-analysis by Dumble, Wolf and collaborators, where they collected all the work that have performed wild crop crosses, and they found that in 30% of the cases, there is a reduction of fitness in F1 hybrids. So I think that this reproductive isolation between wild and domestic forms has been largely overlooked, but could for sure play an important role in the emergence and maintenance of the domestication syndrome. Besides this, if you look at the divergence between wild and domestic forms, they fell right in what is called the gray zone of speciation. So this graph is from a recent publication by Monet and collaborators in 2025. And so what they did in this study is they collected data from 280 plant pairs, either population or species. And they classify them either in the migration category, meaning that they still can exchange gene flow, or the isolation category, they are not able to exchange gene flow anymore. And in the x-axis here, you have the net divergence. So you see that you transition basically from a state where you are a single species to a state where you are two isolated species, and the level of divergence on average is different, right? But in between those two, you transition in a state of semi-isolated species with intermediate level of net divergence. So this zone here is called the gray zone of speciation. And if I plot now the net divergence that I estimated from my wild domestic pairs in the 13 species, these are all the diamonds here, and you see that they fell within this gray zone of speciation. So what I found really interesting is not, well, the idea here is not really to use domestication as a model to study speciation, because domestication is not a speciation process, but rather to use domestication to understand better the setup of reproductive isolation and the setup of early barriers to gene flow with a gradient of divergence that cover this gray zone of speciation. So that's what we are aiming to do. And to do that, we are performing a comparative analysis of the genomic patterns of reproductive isolation in all our 13 species that I described earlier, covering a range of domestication timing, a variety of life cycles, and also mating systems. And the first thing that we did is to develop a tool that we called REACH, for reproductive isolation detection using genomic polymorphism. And what this tool does is that it detects barrier loci, okay? So how to detect barrier loci? So there are genomic patterns associated with reproductive isolation that usually translate in what we call islands of differentiation. And the elevation and size of these islands depend on a balance between selection for reproductive isolation, intensity of gene flow, and recombination. But of course, the pattern of differentiation along the genome is very heterogeneous. It's not only because of barrier loci, right? It's heterogeneous because you have drift, you have variation of recombination along the genome. You also have linked selection that we know can be modeled by genetic heterogeneity of effective population size along the genome. So what should a detection barrier tool do, right? It should model genomic heterogeneity in migration rates, detect barrier loci, so the loci for which migration equals 0. And we would like it to do that even for recent divergence, because we are interested in the gray zone of speciation. And besides, we wanted to provide a comparative framework to allow exploring a gradient of divergence, so that because we have multiple pairs of species with distinct samples and history. Okay, so we need really a comparative framework, while we also wanted to account for the confounding factors that I mentioned earlier, which are demography, variation in recombination, and genomic heterogeneity in NE. So I'm not going to enter into much detail about how this tool works. It was already published. So basically, it's based on ABC, so approximate Bayesian computation, were coupled with random forests. So we explored a variety of demographic models, but also genomic models. And what is important to say is that we run this simulation, but we didn't choose one or the other model. To make it like a comparative framework, we use the hyper model. So basically, we use a subset of simulation, irrespective of the model, that basically fit better the observed data. And with this, what we call this hyper model, then we run an additional set of simulation with and without gene flow to train a second random forest classifier that can tell us for every locus in the genome, that can return for every locus in our genome, a probability of a locus to be a barrier or a non-barrier loci. And so from there, we derived some bias factors that allow direct comparison between species. So what we found using simulation is that divergent time has a strong impact on the overlap between barrier and non-barrier loci. So if you have very recent divergence, like say 1,000 generations, you're not able to tell apart the barrier loci in red from the non-barrier loci in blue. You have no power to tell the difference. You see that the distribution of bias factors are completely overlapping, and you cannot tell the true positive from the false positive, okay? But when the divergence is greater and greater, so if you reach 100,000, then you're perfectly able to distinguish between barrier and non-barrier loci. You have no false positives. And if you're in between, you have some power to detect barrier loci. And actually, with domestication, we are rather in this middle zone. So we were not exactly sure whether our tool that we first applied to much more diverged species, and it worked well, we were not sure how it would do with such a recent divergence as domestication. Or is it staying the same? Yeah. Yeah. So I will show this. I will show this that I related. You will see to the yeah. Okay. So we applied this tool to maize. Okay. So and then to other species, but I'm just showing an example of maize. So we have 20 teosynths, 20 maize. We detect SNPs. We have a recombination map. We perform some simulation. All the simulation, or pretty much 98%, are consistent with heterogeneous migration. With migration, and 67% with heterogeneous migration along the genome. We get our distribution of bias factor. You see it's bimodal, and basically we use this blue cutoff here, and we considered all the loci after this threshold as barrier loci. Okay, so then we looked along the genome, and maize is one of the species for which we know one barrier locus, okay, which is called GA2. And we were quite happy because we recovered this locus, which is known as a prezygotic unilateral incompatibility that prevents gene flow from maize to teosynthase. And so in maize, we found that 3.5% of the barriers occupied 3.5% of the mappable genome. So we run this in, So then we ask whether the barrier loci had a genomic pattern consistent with barrier, right? So we look at statistics such as the DXY or the FST, so the divergence, differentiation. We found that the barrier loci had a greater divergence, greater differentiation as expected. The level of diversity was similar in the domestic forms between barrier and non-barrier loci. Okay, then we looked at the genomic distribution of this barrier loci. So these are the pink traits here. Okay, so they are here shown all along the genome. Then we also detect selective sweeps within the wild form, within the domestic forms. We looked at the overlap between those selective sweeps, known domestication loci, known barrier loci, and the barrier loci that we detected. Okay. So what we found is that there was no significant overlap between selective sweeps and domestication. There was no overlap between our barrier loci with selective sweep in the domestic form or even known domestication genes. So what we are detecting is something else than selective sweeps, basically. And we found that the barrier loci, they tend to cluster more than expected by chance. We actually, if we look at the recombination rates in the barrier loci versus the non-barrier loci, we found that barrier loci tend to be located within regions of low recombination. Okay, so that was true, for example, in the 6 species here. It was true for 5 of them, but you see that the reverse pattern is observed in foxtail millet. And finally, so that's to answer maybe the question of Christian. So this is the percentage of barrier loci that we detected in our genomes, and this is the percentage of barrier loci. So I mentioned maize before with 3.5% of barrier loci. Okay, for 3 species. The simulation were consistent with homogeneous migration, so we did not perform barrier detection. For 2 very recent domesticates, the patterns were so strange that we didn't incorporate them in our study. And so, I mean, this is all of these are unpublished results and also very recent ones. So, but basically, we kind of find a tendency, I would say, between the percentage of barrier loci detected and the time since domestication. So a tendency of increased barrier proportion with domestication timing, but no effect of the mating system. So we could have thought that, for instance, for outcrosser loci, there will be more intense selection for barrier loci than in self-fertilizing species, and we didn't find this. Okay, maybe. Okay, so conclusion of this part, adaptive introgression occurs not only from wild to domestic, but I also showed you that it occurs from domestic to wild, and it can contribute, in the case of Teosante, to the emergence of invasive weeds. We also have shown that barriers between forms can emerge, and that the proportion of barriers tend to increase with domestication timing, but this again is very recent. We have to verify this. We also found that barriers tend to be clustered and located in low recombining regions, and we didn't find any effect of the mating system. So right now, what we are doing is that we have performed crosses within domestic forms, within wild forms, between wild and domestic forms in the 2 directions. We have produced F1 hybrids. We have phenotyped all the hybrids, and it looks a bit like that. So this again is like super recent, and we are not exactly sure what to do with this. So here I'm presenting the data for cabbage, eggplant, common bean. On the left here, dark blue, you have domestic crosses, so between land races. Wild crosses are on the right, the dark red, and in between you have domestic wild and wild domestic crosses. Okay, and you see that often what happens is that in fact the between form crosses are sort of in between the parental crosses, and this is kind of true. I mean, this is the most general situation. So we are not exactly sure now how to analyze this data. We are thinking about it because the original idea was to look for hybrid defects in crosses. So we find some evidence in some species of hybrid defects, but for most traits, we didn't find this. Okay, so I will now end by acknowledging the PI of this project that with who I've been working for several years. So Sylvain Glémin at Ecobio. So Ewen actually made his PhD with Sylvain Glémin in Ecobio, and I was co-advising him. Catherine Dogimon at GAFN in Avignon, and Yves Vigourou in UMR Diade in Montpellier. And I would like to thank, of course, the work of Ewen and Arthur, but also the work of Tenaillon, which was hosted in Yves Vigourou's lab and did all the genomic, analyzing all the SNP discovery. And so it was really a huge amount of work. and Josefina Wollfeiler, who was a postdoc at GAFL with Catherine de Gimont. And so she did analysis on RNA-seq data that I did not present today. And finally, I would like to thank the people in my group, and more particularly the ones that have contributed to this research. So Domenica Manicacci, Harry Belkram, Pierre Vérard, Karine Alix, Agnès Rousselet, and Martine Guillou. Okay, so thank you for your attention.